thank you very much. Welcome everybody. So the title's a little bit misleading because I just thought that I'll try and make it as boring as possible and I can take it easy. So I've been living in Asia for about 15 years, most of the time in Hong Kong, as Chris mentioned, and recently in Singapore. And one of the major interests that we have is emerging infectious diseases, zoonotic infections. In terms of influenza and indeed a lot of the other diseases, such as Nipah, SARS, a lot of them have emerged out of that region, and it's basically why I continue to live there, because this is far, far too far away. Okay, so a zoonotic disease. So as all good scientists do nowadays, rely on Wikipedia. So I just picked up the quote from, or the definition from Wikipedia, which is not bad, but it's an infectious disease transmitted between species, sometimes with a vector from animals other than humans to humans, or from humans to other mammals. And, but really the reverse is the humans to other animals is never really discussed because we don't care if they get sick. In terms of zoonotic diseases, there's many. There's really good resources available from the Centre for Food Security and Public Health, which sort of summarise everything. They've got a big list of what they consider to be the major zoonotic agents. Of course, a lot of bacteria, fungi as well, but I'm going to be concentrating on viruses today, and in particular influenza, because it's the only thing I know about. So this is just an example of 4 different diseases and the interactions of multiple different host species and the environment, soil or unknown environment, and in the emergent pathway for infection to humans. And this is the major focus of the bulk of the research that I do. I'm going to go into and explain about the farming systems and the poultry marketing systems that exist in Asia and how they contribute to the emergence of different influenza viruses as human pathogens. In terms of zoonotic infections, the sources of infection, this is, was summarised by Jones and friends in Nature. So we have basically, these were just reports of infection, but they're individual cases, so that's why Europe looks like it's going crazy there. But you can see generally there's been an increase in new zoonotic infections, whether or not that's due to actually there is more infection or reporting, probably a combination of both, and largely global in distribution. So it's not something that is necessarily restricted to a single area. And then they also wanted to make some sort of prediction for different types of infections and where they may came from, and looking at hotspots. So generally with outbreaks, so we have an outbreak of a certain disease that infects humans. H7N9 recently in China is a very good example. So you get this outbreak, everybody's super surprised for some reason that they've had this infection come out, and then we kill the animals that are the infection source. And this happened in SARS, this happened with Nipah in Malaysia. But very little is done to actually try and understand the systems, the ecological systems that these viruses emerge from. And rather than continually pretend to be surprised and then kill all the infection, the source animals, it's probably a lot better to try and find out something about the systems that they emerge from. If you understand the system, then you can at least do something about it. If you know that there's interaction between viruses, interspecies transmission between different viruses, and I'll go into this later. So it's not just the species barrier at the human-animal interface, but it's also between the different animals that are in these systems. So generally we depopulate, and vaccination is also used as a control, obviously not as a response necessarily. And if I have enough time at the end, I'll also go into vaccination with regards to influenza. And as I said, it's the only thing I know about, so that's what I'll talk about. So key species involved with influenza. So you have wild waterfowl and other species. The waterfowl are sort of considered the main reservoir. They keep on finding more now. There's 16, 17, I think there's 18 and 19 hemagglutinin and subtypes now. So the last two come from bats, and they've not been found in ducks. And the same with the neuraminidase. Poultry, and particularly chicken and duck, act as another large but also separate source of influenza viruses. And pigs, which are important for humans. So pigs, the 2009 pandemic virus, that came from pigs. I think the 1918 one did as well, but people argue about that. But the other interesting thing about pigs is they're very susceptible to human influenza. So there's this constant reverse transmission back into pigs that doesn't really seem to establish. But very important because of the similarities between the viruses that are carried between pigs and humans, and of course humans. This is probably getting a little bit tired now, but you have also have aquatic mammals that do have some spillover, usually from well-intentioned people. There were some people working on seals in the west coast of the US, and then the seals ended up with pandemic H1N1 influenza that probably came from the people that were working on them. You have this sort of dead-end lineages in horses, domestic poultry, where there's a lot of transmission between the wild waterfowl to the domestic poultry, and then to humans, and between pigs and humans. So this is increasing now, and sort of this perception sometimes that, you know, we've had this recent pandemic, we had this H7N9, and there's these big increases in the surveillance that gets done, and you think, okay, well, it's all going to be right now, because generally, if asked, people will say, in terms of prevention, what do you need? You need more surveillance, because you need to know what's there. But you get these little spikes in surveillance, and then nothing. So in terms of long-term influenza surveillance in Asia, there's one program. In terms of long-term influenza surveillance in pigs, there's one program. Since the pandemic, effectively nothing has changed whatsoever. In the US, it's changed a little bit, but there's also not a lot of effort is made to understand to look at the interaction between animals or humans. So it tends just to be looking at one or the other, but that is changing. So just using H5N1 as an example, I don't know how much of a background I need to get. H5N1 first emerged in humans in 1997 in Hong Kong. It infected, I can't remember how many people, and it killed 18 of them. Or did it infect 18 and kill a subset? Can't recall. The reason that this bothered people a lot was because up until that point, it was absolutely 100% known that avian-type influenzas did not infect humans directly, that it had to come through some sort of mammalian intermediary, such as a pig. So just a few years before that, there were people that were taking multiple different subtypes of avian influenzas and doing infection studies on individuals, and just kind of shocked, but apparently they got approval for it. So this was the first example of a direct infection from avian to a human, and it really concerned people because it also had a very high mortality rate, and clinically it's not a pretty disease. The other problem with H5N1 is that it's actually a major panzootic, and I've checked, and apparently that's not a made-up word, where something like 400 million birds have been killed, and there's been economic losses of $20 billion, apparently, in terms of killed animals. So even disregarding this number, one of the major problems with this as it pertains to global health is that this is all protein out of the mouths of populations that are probably least able to afford having reduced protein. This is an old map from 2008 that I guess showed H5N1 at a major extent. This doesn't necessarily mean there was infection here. It's coloured by country. But Indonesia is a major area of influence of H5N1 activity, all through Southeast Asia, China in particular. And now out of all of these, India's still got a large problem, and Egypt. The other areas around here, not so much anymore. This was a more recent, so this is from February to August of 2013, and this is where there's been recorded outbreaks from. Of course, it depends how good the surveillance is. But again, sort of clustered around Indonesia, parts of South Asia, and in Egypt, where it is really a serious problem. So, one of the remarkable things about H5N1, so H5N1 is what we call a highly pathogenic avian influenza virus. So this has a different molecular, is that this making that more?
Lower it, just lower it a little bit, I think you're.
Yeah? More?
You speak loudly anyway.
Hmm?
There you go.
I've got a gentle voice, what are you talking about? Okay. Now, where was I? So, yeah, one of the remarkable things about the H5N1 virus is there's been other highly pathogenic viruses before. There's a fairly long history of them. They tend to be restricted to certain subtypes, H5 and H7. What's really unusual about this is that it's persisted. It's continued on for over 10 years of a single hemagglutinin lineage. And it's also has diversified a lot. So normally you get an outbreak and it'll burn through the poultry flocks and then it's gone. So a problem with the H5N1 is in terms of vaccination and control. So in poultry, vaccination is used as a major form of control. So because the virus has become endemic in the chicken populations, chicken and duck populations, the only way to protect the chickens is to vaccinate or to keep them disease-free, but that's impossible. Ducks generally don't get sick from these highly pathogenic. It's just something that happens in the land-based poultry like chicken. So the coloured triangles represent clades of H5N1 hemagglutinin that are still circulating at the moment. And a problem is that there's not a lot of cross-reactivity when you look at the vaccines for these different viruses. So even within here, the naming system's a little bit ridiculous, but viruses down here might cross-react with one another or cross-neutralise, but it won't protect against here. And these different lineages, because the viruses have been moving around, they now co-circulate together as well. So you either have to have a multivalent vaccine or something that's broadly cross-reactive. Another problem with H5N1 is that you get a lot of reassortment. So influenza, it's a I just completely got that wrong. It's a 8-segment genome. It's a segmented RNA genome. And if you get co-infection, which increasingly now there seems to be, we used to think it was reasonably rare, but there's evidence now that the co-infection is incredibly common. If you get co-infection of 2 different viruses, they can swap genes, reassort, and then you end up with these hybrid progeny. So it's kind of like recombination, except it's with the whole gene segments when the viruses are budded off. And with H5N1, over time, you end up with all these different gene segments. All these different reassortant viruses. They have the same hemagglutinin and the neuraminidase, but the internal genes change a lot. And there's also you get these ones, these little plus numbers, other genotypes that were detected, but they didn't last for 1 year. So you have this big pool of diversity that is generated. So one of the interesting things that is becoming apparent now is that that there may be some linkage between reassortment and antigenic change, and in particular these bursts of antigenic change. So the fact that you have a diverse hemagglutinin, and a lot of the diversity in hemagglutinin is because the virus is spread out to different parts of the world. So when the virus was initially picked up in Indonesia in surveillance in 2003, it was concentrated around Java, where the bulk of the poultry production is centred. And then after that, you see it spread out to Sumatra, and then you out to the, I can't remember my directions, west of Sumatra, and then east through to Sulawesi. Indonesia's fairly heavily populated, and it's got an awful lot of islands, and you start to see diversification within those areas, because once it gets into Java and spreads out, there's not actually a lot of mixing between the bird populations in the different animals, because they get eaten, right? So in terms of what generates the diversity for the influenza, it's very easy to look at substitution rates, and you have frequent reassortment, and then you might have selection pressures as well. But really, the ecology of the virus and the host are what's going to be driving a lot of this diversity, or generation of the diversity. So these are some fairly boring slides of text, but I'm just going to describe what happens anyway, so you can read or not. So there's a lot of poultry. There's huge amounts of poultry. One of the interesting things in China and Southeast Asia in particular is that they have polyculture. So they'll have multiple birds being raised, multiple different types of birds being raised together. Now, this might not seem to be a problem, but it has shown to be a problem. So we have aquatic birds, so duck mostly, and then land-based poultry, which is chicken, quail, these things called chukka, pheasants, pigeon. All of these different things are sold in the markets, right? They're farmed and they're sold in the markets. Pigeons, which have been fairly benign when it comes to influenza to the moment, have been implicated in H7N9, so they were found to have some H7N9 in them. Another problem is that particularly in China, there's now large farming operations near major aggregation sites for migratory birds. So this is obviously a map of China. This is Qinghai Lake. There's a nature reserve, and there's a very big lake. So they have big duck farms on the shores of these lakes, because it's a source of water where they can farm their ducks. But there's not high biosecurity. So you also have, this is a major aggregation site for migratory species, and you have a high level of interaction between them. And the thing to remember about influenza is that with the life cycle, it's basically a gastrointestinal infection of the birds. So it comes out in their feces into the water, and then it infects through that cycle. So this, you know, there's a lot of evidence, or there's been a lot of work done showing that it survives very well in water as well. So you have these wild birds come in, and then they're going to be able to transmit, and vice versa, viruses from backwards and forwards between the two populations. This is also a fairly typical setup for farming of ducks. So I don't know if anybody ever heard of there's duck herders. So there's actually flocks of ducks that get walked up and down the length of places like Vietnam. I don't think it's so common in China. But the ducks are fed on the rice paddies, and it's very common. It also makes control a little bit difficult because the ducks keep on wandering off. So the other component of where these viruses sort of hang out is the poultry markets, and these are probably the main problem when it comes to trying to control this virus. And I guess before I go on to this too much, I want to say that in terms of finding out what the systems are, and if we inform ourselves about the ecology of the production and the marketing systems of these birds, almost nothing has been done on it. It's really, really difficult. If you're sort of walking around, in a market, with a computer or taking notes or taking pictures, then you'll probably find yourself run out of there fairly quickly. So, poultry markets. The main problem is that there's multiple species have been housed together and they've got huge viral loads in feces and drinking water. There's also frequent movement of birds in cages between different regions and different farms. So there's no, typically how it works is you have either individual farmers, like small farmers or larger farmers, they'll bring their birds to a central market, a wholesale market, and then the people will buy their chicken for the day, and then they go off, right? And then sometimes the cages go, sometimes the cages go back. So it depends on the relationships between the people. And then when they go to the market, then those cages might be brought back and filled up again. But it's sometimes they're just like the shipping crates that they have, the wooden ones where you don't necessarily keep your shipping crate the entire time. And these things are very, very good vectors because they've got fomites on them and that are full of viruses. And that's one of the major infection sources. And these live poultry markets, so you have your central wholesale market, and then you might have multiple retail markets within the city or within a metropolitan area. And for instance, in Hong Kong, the chickens that were getting sold in Hong Kong, they come from an area that across the basically sort of this area. So Hong Kong's down here somewhere. So we'll get birds from Funan, Hunan, Yunnan, Guangdong, and I've forgotten what those two provinces are. And the same with the pigs. So they're drawn from these very large areas. So you basically get a mixing of poultry from different regions and different origins, and it can really reduce the efficacy of the control effort. So you might have one particular area where they have really good biosecurity, really good control, but then everything gets chucked together and then it all goes out the window because they're being mixed in together with this population that was raised with very poor biosecurity. So once they go through because the infection doesn't take that long, once they're sort of into the markets, then they can be shedding virus. So even if you manage to eradicate it by adopting comprehensive control measures in the affected region, you could actually then get transmission back anyway. So these are just pictures of what different markets look like. So these here is ducks. Obviously we have chicken, and then there's that looks like duck there as well. So the problem with having different animals mixed together is a good example of what happened with the 1997 H5N1 virus. So that virus, you had the H5 and the N1, that obviously came from a progenitor virus. But then the internal genes came from H6 and H9. What they found when they looked was that the H6 and the H9 viruses, they weren't so much in the chicken and they weren't so much in the duck. But when there was lots of them in quail. And then for some reason, when the H6, the H9, and the H5 were all put together in these, because you had chicken and quail housed together, then they seemed to reassort really well and make progeny that were very good at killing people. But once you remove the quail out of the system, then things sort of went back to normal. So if you put quail in, they had H9, but originally the H9 does come from duck, but they just seem to be well adapted to it, or it to them. And so if you have these multiple different species, you end up getting some sort of host restriction in them. and they harbour different types of viruses, and then they'll also diversify in that population itself. So it tends to find if you remove out other hosts, then there's less risk of actually getting an outbreak, and less risk of getting reassortants, which could then be vaccine escapes from the current vaccines. And again here, so you have the birds stacked on top of one another. It's not unusual to see multiple species, so you might have, you know, chickens here and duck here in the same. It kind of depends on the structure of the market, but again, this hasn't been looked at in any detail, whether you have, you know, just a chicken-only vendor, or you might have a chicken and a duck vendor and things like that. This is a market in Hong Kong, which has recently at least had a very good record of no H5N1 in the market. So what they do there now, in order to control H5N1, so you're not allowed to have quail at all. Initially they separated them, but there's just not allowed into the markets. Then they had one clean day a week where everything had to be steam clean. And after that, they said that you'd have to depopulate at the end of the week, so you couldn't carry birds over in the market for the weekend. Now you have to depopulate and steam clean at the end of every day. And I think eventually they're phasing it out, which is a pity, because the freshly killed chicken from here tastes really nice. So, the vegetarians. But this surprised me. So these were some photographs I took a while ago, but that looks like a pigeon. And I didn't realise that they still allowed multiple species. So this is just a close-up of the cages. So these are actually quite clean. So that's the close-up of the cages there. And as you can see, you know, this might look, it's not that clean, but it's pretty good. So you chop their heads off and put them there to bleed them. And then this is where you wash them out. And then this is all the gizzards that's been soaked, and you can see bits of chicken carcass and stuff in there as well. This is a very clean system, right? But you can see if things weren't maintained well, and if it was on a larger scale, that it would be quite easy. It would be difficult to keep clean, and it would also be difficult to control infection there. So I think I've already talked about that in terms of the transmission and outbreaks, and how they led to the diversification of the virus. So in terms of influenza viruses in general, so you have this gene pool of wild duck that has these low path viruses. So low path are your everyday non-killer ones, but they have H9N2, which has caused human infection. That's low path. So the low pathogenic avian influenza is actually defined by just this marker. So basically, and it's defined for what it does to chicken. It's not defined for what it does to humans. So a low pathogenic virus, it has a fusion a cleavage peptide of the hemagglutinin to allow infection. It's restricted to an enzyme that's in the upper respiratory tract. You get these additional residues at that cleavage site, and then it can be cleaved by ubiquitous. So you get a ubiquitous enzyme, so you get systemic infection of the birds, but it's always defined for birds. And then these domestic duck sort of act as a gene pool between the low path and these high path viruses in the farms and the markets. And so at places like Qinghai, you get the low path viruses come in, and these are brought into the ducks, and then because the ducks are also infected with the H5N1, then they get mixed around, and then eventually these viruses find their way into the poultry as well. And then once they get into the poultry, they've got new genes in them. The new genes they can induce, if it's reassortant, you can end up with antigenic change, as well as just because there's the regular evolution, and then your vaccines aren't working anymore, or you end up with multiple strains. I can either go into vaccines or not. How are we doing for time?
Pretty good to be finished up around 10 till to give time for questions.
Okay. So generally, so in terms of vaccines, I'm just going to talk about vaccines in terms of the H5N1 again, and how it's being used as a control strategy. So for avian influenza, there's different reasons for using vaccines. One of them is to stop people from getting infected, and that's been used quite well in places like Vietnam, where they had a lot of human infection, and then they started vaccinating. It reduces the overall load in the poultry, and then less people get sick. In other places, they've tried to approach it for eradication, which hasn't worked, quite frankly. Generally, if you have an outbreak that's controlled by culling, you get eradication is achieved within 1 year. But it comes with a cost. So Thailand did this. They don't use H5N1 vaccines in Thailand because Thailand is a major exporter of chicken. And if you've got H5N1 vaccinated, then a lot of countries will not accept the poultry because it could, it's not going to provide sterilizing immunity, so they could have some infections that wouldn't be detected. You'd have to do a lot of testing and everything else. And non-vaccinated chicken is great. I mean, it self-controls because the birds die, right? They die very quickly, and it just burns itself out because it is so pathogenic in them. And generally, you can achieve eradication within a year. So what they did in Thailand was if they have an outbreak, then depending on, I think it changed with time, but they would have these sort of cull zones around the outside, okay? And then everything that was within a certain radius of the outbreak, all those flocks would be depopulated or killed. There's a problem with doing the depopulation because if you don't actually give money to the farmers for lost income, then as soon as they find out that a neighbour's farm is infected, they'll get their birds to market as quickly as possible. So generally, eradication needs to involve some component of compensation. And then so that's just with culling alone. It usually works fairly quick. If there's been a combination of culling and then with vaccination, generally the eradication, if at all, has not been as swift. And there's also been instances of vaccine escape or failure in poultry populations, and this is increasingly becoming a problem. So specifically in China, so most of the work that I did on influenza was in China, and we had an extensive surveillance network across the sort of I think the 6 southern provinces of China, covering a human population of about 500,000. So in China they tried doing large-scale vaccination, which they're also they're still doing actually, to try and eliminate or manage H5N1. And quite amazingly, there's been 35 billion doses of the vaccine administered across those years, but you still have H5N1 prevalence. But also the coverage of the bird population being vaccinated is important. So you need to make sure that you're reaching a certain level of coverage for the vaccination to be effective, unsurprisingly. So the 35 billion doses of the vaccine sound really impressive, but China has an annual production of 15 billion poultry, right? 15 billion poultry. So that's what's being produced and sold every year. So that's you've got all the other birds in the production line behind it, and 70% of that population is still reared in small-scale farms or in backyards. So if you've only got 8.75 billion doses annually, it's not going to cover the 15%. So the large numbers of the unvaccinated poultry would remain capable of spreading virus and through poultry movement and the live poultry markets. And a lot of the vaccines don't actually give protection, sterilising immunity or anything like that. So even if it is vaccinated, a lot of them are 2 shots as well. But the 2-shot vaccines, as I'll see, this is a rather long one. They have these 2-shot vaccine regimes, but which work wonderfully in the laboratory. But by the time you put it into the real world, the 2nd shot is given after the bird is dead. Because they don't match up with the production cycle of the animal itself. So it looks great, but on a practical level, it's not going to work. So one of the problems in particular is ducks. So ducks are like the silent carriers for H5N1 and most of the influenza. So they don't, as I said, the high pack is defined for chicken. In ducks, they're generally not susceptible. And the ducks are also highly refractory to vaccination. So if you vaccinate them, you don't get much of a response. And there's a significant number of ducks, and they tend to harbour the virus as well. So the other, problem that I have with vaccination, when it's done on a large scale in chickens, is that there's not much discussion and certainly very little research on the effect of vaccination on the evolution of the virus in terms of driving potential antigenic change. It's really difficult to get information necessarily about what antigen is being used in a vaccine, what the dosing is like and everything else, because a lot of it is done within, it's within large production companies, and it's not the type of thing that they're keeping. Not the type of information that they would typically disclose. So in terms of where they've, and this is very tenuous, but where you have multiple antigenically divergent sublineage emerge regularly is in China and Indonesia, and then more recently in Egypt. And what all of these have in common is that they vaccinate extensively. And if you don't apply blanket vaccination, then they've also not had the virus established endemically. So it kind of comes down to a decision of, do you want all the birds to die, or do you want to keep them, and then you're going to have to live with H5N1 in the flocks on the long term. So this is not necessarily a criticism of vaccination, just that there's consequences for whether or not you do or don't vaccinate in terms of control, but in terms of eradication, then the answer is probably going to be no when it comes to vaccination. So there's an example of H5N2 outbreaks in Mexico, where they've, in the 95, 98, so they're vaccinated with these are low-path viruses, but there's been continued circulation for 20 years. And they've also shown, so this was one of the few studies where they've actually done this work, they've also shown that the use of the vaccines has led to antigenic drift and then diversification in those viruses away from the vaccine strain, and then you get an emergence of vaccine escapes. But one of the things that's never been looked at is the effect of large-scale vaccination on H5N1 in these different countries. I tried to convince my old boss that we should set up our own little pretend chicken farm. Not chicken farm, chicken market, like the ones in Hong Kong, so we could have them, all the cages lined up, and we could infect one bird there and then sample across and then sequence everything, and we could have vaccinated. But he said no, I wasn't interested in it, but it would have been fun. And so that's it. Funding from NIH and Duke NUS, A-STAR and Ministry of Health, some Hong Kong funding, and Li Ka-shing Foundation. Thank you.